Now, the last day we completed the concept of genetic code, and we got to know that it was the experiment of Nirenberg and Codon that actually brought out the relation of the 64 combinations of the genetic code into 20 amino acids. So how the 64 combinations of the code is related to 20 amino acids, we got an idea of that in the earlier class, and we also saw certain important properties of the genetic code. Now, with this, we come to the start of our translation machinery once we are aware genetic code is. Now, when we talk about translation machinery, the translation machinery, as I said, is a multi-complex process. It involves a ribosome, it involves mRNA, it involves the tRNA, and most importantly, it involves the amino acids. So when we talk about translation, it is a number of molecules coming together in the cytoplasm to bring about a coordinated action, the output of which is the production of a polypeptide. So now this is in a nutshell, what are the different components required when we talk about protein synthesis in bacterial cells. We will restrict ourselves to protein synthesis in bacterial cells, even though there is much similarity between protein synthesis in eukaryotes and prokaryotes, but the initiation of eukaryotic protein synthesis is somewhat different from prokaryotic protein synthesis, because if you remember, prokaryotes do not have a 5' cap and a 3' poly A tail. But since eukaryotes have a 5' cap and a 3' poly tail, so the protein synthesis is the initiation process is somewhat different in prokaryotes and eukaryotes. We will not be getting into the eukaryotic translation machinery. We will restrict ourselves to only the prokaryotic translation machinery. So the basic 3 stages in which a translation cycle can be divided is the initiation, elongation, and the termination. So when we enter these 3 stages, we enter directly into the translation machinery. But prior to that, there is another important thing that needs to be done so that this process can start move smoothly. As you can see, prior to that, one important thing that is to be done is the binding of the amino acid to the tRNA. If you remember, when we talked about Ubel's hypothesis, I had showed you that there is a tRNA which via its anticodon recognizes the codon on the mRNA. And the tRNA actually carries the amino acid at its, if you have that picture, let's have that picture just a second so that you can see that once more. So, and just a second. As you can see, so this is the tRNA I was talking about, and this is the anticodon of the tRNA, which is read in this direction, 5', 3'. This is the codon of the mRNA, read in this direction, 5', 3'. And you can see the tRNA is carrying an amino acid at its 3' end. So the first part, that is prior to initiation of protein synthesis or during protein synthesis, whatever, because this is a continuous phenomena. Because whenever a codon is recognized, the relevant tRNA is brought into action. It binds with the relevant amino acid and is then inserted into the decoding center of the ribosome. So this is an important part which occurs simultaneously along with the initiation and elongation process of translation. During the initiation process, this starts, but this continues simultaneously along with the elongation process. So, as I was saying, let's get back to the slide once more. So, as I was saying, so when we talk of binding of amino acids to tRNA, we see that what are the components involved there? The amino acids, the tRNA, amino acid tRNA synthetase. I'll talk about this today. Later on, this is an important enzyme. And as you can see, the function, it attaches amino acids to the 3' end of tRNAs. And obviously, the reaction is ATP dependent. When we move into the initiation phase of translation, we require the mRNA, we require the initiator tRNA, we require the 30 and the 50S. So these are the ribosome. And then we require certain proteins which behave as initiation factors. When we move on to the elongation phase of translation, so this is again the ribosome, which is now known as the 70S initiation complex. And we again require charged tRNAs. What are charged tRNAs? That is tRNAs carrying amino acid at their 3' end. They are known as charged tRNAs. Then we have a new set of proteins known as elongation factors. When we started, we had initiation factors. Now we have elongation factors. And interestingly, you see there's a peptidyl transferase center, which helps in creating the peptide bonds, but this is a part of the ribosome itself, okay? And finally, when we come into termination, is the release of the mRNA from the ribosome, release of the polypeptide from the ribosome, and the opening up of the ribosome. So that is again facilitated by certain proteins known as release factors. Before getting into the details of this step, as I said, the basic raw materials required for translation is mRNA itself. Then we have the tRNA. Then we have the ribosome. And most importantly, we need this enzyme, amino acid tRNA synthetase, to charge the tRNA with their cognate amino acids. So we will discuss the raw materials first in a nutshell, and then we will enter into this particular process step by step. So when we talk about the first raw material, that is mRNA. When we talk about mRNA, during transcription, I showed you that if I look at the transcription unit, if you remember, I told you, if I look at the transcription unit, the mRNA has a 5' leader sequence, then it has a protein coding sequence and a 3' trailer sequence. So the protein coding region is decoded into the polypeptide. There is no relevant protein part of the 5' leader sequence, and there is no relevant protein part of the 3' trailer sequence. So in a, where did my cursor go? Just a second. I have lost it again. Oh, sorry. Yeah. So if I look into an mRNA, if you remember, we have a 5' end where there is a small cap sitting. Oh, sorry, this is a prokaryotic machinery, so there is no cap present. So let's remove this cap. So we have a mRNA, we have a 5' end. If you remember, this is something we did in our transcription class. And we have a 3' end, right? And then, as I said, there is a region of the 5' end which is not decoded. So this is the 5' leader sequence. And again, we have a region in the 3' end, which is again not decoded. This is the 3' trailer sequence. So this is the protein coding area. So this much is the protein coding area of a mRNA. So this is a transcription unit. And when we look into the mRNA, what did I say as a property of the genetic code? This protein coding region, so this region, I'm talking about this protein coding region, it contains non-overlapping string of codons. If you remember the property of genetic code, so it contains non-overlapping string of codons known as open reading frame. So this is another new term you learned today. So this area, the area of the mRNA, which is containing a string of codons and which will be coded into our polypeptide, is known as the open reading frame. Each open reading frame specifies a single protein which starts and ends at internal sites within the mRNA. So it will code for a single protein and it is starting here, which is, this is the actual position from where our mRNA is starting, but this open reading frame is starting at the internal site. It is starting at the internal site of the mRNA and also ending at the internal site of the mRNA. The second property of the mRNA is that the mRNA also clearly points out from where this open reading frame is started. In bacteria, as we studied the earlier day, the initiation codon is AUG. Okay, so that is our start site for the open reading frame. However, in some bacteria, it has been seen that the start codon, this is in case of bacteria I'm talking, the start codon might be 5' GUG3', or it might be 5' UUG3' also. That are exceptions. But the universal rule is that the start codon is AUG. In eukaryotic cells, it is always the 5' AUG3'. So this is from where the protein coding is being started. So this is the one boundary of the open reading frame. And the other boundary of the open reading frame, if you remember, are the presence of the stop codons. So the other boundary, we have 3 of the stop codons, any one present. So for this example, let me write UAA. So this is a stop codon. So every open reading frame has a start codon and every open reading frame has a stop codon. And this is the mRNA, and this open reading frame will be translated by the ribosome to give us a defined polypeptide. The AUG, that is the start codon, this ends from the N-terminal end of a polypeptide, and the UAAN, that is the stop codon, forms the C-terminal end of the polypeptide. The presence of AUG, in fact, informs the ribosome. This is where you start. This is the first amino acid, and then you go on synthesizing the amino acids, taking 3 nucleotides together as one codon, and accordingly build up a polypeptide chain. So the start codon actually defines the reading frame for all subsequent codons. So now when we are thorough with open reading frame, let us have one more concept here. In case of eukaryotes, eukaryotic mRNA usually have only one open reading frame. That is the one I've shown you here. We have a 5' leader sequence. We have a 3' trailer sequence. And you can see this is a single open reading frame, which starts with a start codon, which ends with a stop codon, and in the process, a single polypeptide is being produced. So, mRNAs with a single open reading frame are known as Monocystronic mRNA. Monocystronic, where cistron is equivalent. The word cistron is equivalent to 1 gene. So the concept is 1 gene, 1 polypeptide. Why 1 gene? Because a single gene gave rise to this transcription unit. And from this transcription unit, I am getting a single polypeptide. So the hypothesis is 1 gene is giving rise to 1 polypeptide. And this single open reading frame is considered to be a cistron. The 1 gene is also considered to be a cistron. And this kind of mRNAs are known as monocystronic mRNA, which is the characteristic properties of all eukaryotic mRNA. But there is always a but. When I say a but, there is something else to it. Let me draw another mRNA. So as I said, we have a 3' tailor region, which cannot be translated. So let me draw the 3' end. So this is the 3' end, right? And then we have a 5' end leader region, which is not also again translated. Just now we had this concept. So we have the 5' end. So this is not the 3. This is the 5' leader sequence, and that is the 3' trailer sequence. Now let us imagine that this segment is divided into 3 parts. See, this is the common leader, and this is the common trailer sequence. Okay, but as I told you, there is a single open reading frame. That is, this particular coding region is coding for one polypeptide. Here what we are seeing, this particular region has been divided into 3 defined segments. Each segment is coding for a polypeptide with different function. So what do we see in this picture? We are seeing that a single mRNA having a common trailer region and a common leader sequence and a common trailer sequence is having 3 different segments in the coding section, and each segment is coding for a different polypeptide. So just now I said the concept was that one gene is giving rise to one polypeptide. A gene is equivalent to a cistron. Interestingly, in bacteria, this was seen. So if this is one cistron giving rise to one polypeptide, then this mRNA has 3 cistrons embedded in its coding section. It has the information of 3 cistrons embedded in its coding section. 1 cistron, 2 cistron, 3 cistrons. So it is having the information of 3 cistron embedded in its coding section. So this kind of mRNA is known as Polycystronic Mrna, where the coding region of the mRNA has 3 open reading frame. You can see there are 3 open reading frame. 1 open reading frame, 2 open reading frame, 3 open reading frame. Remember, an open reading frame is you start with a AUG and you have a stop codon on there. That's the open reading frame. Okay. So this mRNA has a common leader sequence, a common trailer sequence, but 3 different open reading frame juxtaposed, put together. And these 3 different open reading frame are giving rise to 3 different polypeptides. So this is a polycystronic mRNA. So we developed this concept of what is a monocystronic mRNA and what is a polycystronic mRNA. Now a question that automatically might have come to your mind is that if an open reading frame is symbolized by a start codon and a stop codon, how does the ribosome understands that this is one gene? So when I come to this area, I have to stop decoding this segment and start decoding this segment. So whenever ribosome comes into this particular area, the ribosome has to realize that I have to end the synthesis of the earlier ORF, and I have to release that polypeptide and start synthesizing the next ORF. How does the ribosome read this? So what is seen here is that in these junctions between the 2 ORFs, the sequence that is present is usually A, U, G, A. I should give a direction because whenever we talk about sequences have defined direction. So it's 5 prime, A, U, G, A, 3 prime. So whenever the ribosome is coming here in this particular area, in this junction point, it is coming. It is reading a sequence like this. So you can see this sequence Let me change the pointer color in color. Let's make it blue. Okay. So you can see this particular sequence has a start codon embedded in it and also a stop codon embedded in it. So AUG is read as the start codon for the second ORF, and UGA is read as a stop codon for the first ORF. So at that junction, these sequences stay. The junction between ORF1 and ORF2. And at this junction, the ribosome reads the sequence as 5' AUGA3'. AUG acts as the start codon for the second ORF. UGA acts as a stop codon for the first ORF. So when ribosome reads a sequence like this, what it does, it releases the polypeptide that it had made as a result of decoding the first ORF, and now starts decoding the second ORF because it has also read the AUG start codon. So this phenomena of linked translation, so translation is linked between two ORFs, as you can see here. So this phenomena of linked translation between overlapping open reading frame is known as translational coupling. So this is the concept of translational coupling, where there is a linking between the translation of two open reading frames. So we have this basic concept of what is a monocystronic mRNA, what is a polycystronic mRNA, and the concept of translational coupling. And we understood that whenever we look at a mRNA, as I said, the open reading frame is bordered by a, let's look into this picture now, is bordered by a start codon, and then it has a stop codon. The ORF is read and it has a stop codon. So if I draw an mRNA, let me change the color of the pointer, just a second, so you can understand. Pointer options. So let me now draw, let us pointer options, pen. So suppose this is a mRNA. What happened? I changed the color, pointer options in color, red, pointer options, erase all. Now let us imagine this is the leader region of an mRNA. As I said, the leader region of an mRNA, the leader region of an mRNA, just a second, is not translated into a corresponding pointer option pen. Penta gelo kotha. Okay. The leader region of an mRNA has a 5' end, and then it has an open reading frame, and then again we have a 3' end. So let us concentrate on a monocystronic mRNA. Let's come out of that polycystronic concept. So as I said, the open reading frame, as you can see here, is bordered by A, U, G at the 5' end, and it has a stop codon at the 3' end. So if this is the 5' end, we have a 3' end on the other side, and this is a stop codon. So let me put a stop codon here, fine. Now, the mRNA, as you can see, is providing one information to the ribosome, that AUG is the initiation codon, and it is from this AUG that you, the ribosome, should translate or decode the information into a polypeptide. But this initiation codon is not sufficient enough to indicate the ribosome to exactly point out where to bind. Now, in case of prokaryotes, there are two subunits of ribosome. The 30th subunit, you know all this. You have studied it in class 12, that the ribosome in prokaryotes can be divided into the 30th subunit and the 50th subunit. The 30th subunit is a small subunit and the 50th subunit is a large subunit. So as I said, the 30th subunit actually binds first to the mRNA and then only the 50th subunit binds. So the AUG initiation codon is not sufficient to tell the 30th subunit that where should you bind the mRNA. It has been seen that a sequence upstream of the 30th subunit, which is known as the a sequence upstream of the AUG sequence, a sequence upstream of the AUG codon in the mRNA is called the ribosome binding site, and this sequence is necessary for the 30th subunit to recognize the mRNA and bind to the mRNA. This particular sequence was discovered by John Shine and Lynn Dalgarno and is a purine-rich sequence. So this sequence, as you can see, has been shown here, reads in the 5' direction, AG 5', AGG, AG 3'. And this sequence can pair with a complementary pyrimidine-rich sequence. You can see it's pairing with a complementary pyrimidine-rich sequence of the ribosomal RNA that is present in the 30S subunit. So what happens? So we have this sequence 8 to 10 nucleotides upstream in the ribosome. That's what I said now. And this sequence, so we have this sequence somewhere around here. So let me change, I've done it this color. So this sequence is there. And in the cell, there is this 30S ribosomal subunit. So this 30S ribosomal subunit, it comes and binds to this portion of the mRNA. Within the 30S ribosomal subunit, there is the ribosomal rRNA. The ribosomal rRNA complementary base pairs complementary with this 30S ribosome, with this mRNA via its own pyrimidine-rich sequence, UCC, UCC 3'. So from this particular information that we got today, the mRNA has two vital points which helps the ribosome to recognize the mRNA. The first vital point is the presence of the ribosomal binding subunit, the Shine-Dalgarno sequence. So the first one is the Shine-Dalgarno sequence present in the mRNA that actually tells the 30S ribosomal subunit exactly where to bind. Within the 30S ribosomal subunit is present the 16S ribosomal rRNA. So the 16S ribosomal rRNA binds with the Shine-Dalgarno sequence via its complementary pyrimidine-rich region. And then the second point of importance on the mRNA is the initiation codon. So once the mRNA has got, the ribosome has got itself attached to the 30S ribosomal subunit, the second information the mRNA is sending to the ribosome is where the ORF is starting. That is, from this point, you are going to translate and make a polypeptide. So as you can see, so the mRNA is providing two vital information to the ribosome for one, to sit on the mRNA, second, to start the translation process. So just, I just want to show you one picture that is relevant to this. That picture is just a second. At times this hangs. So this is the 2 ribosomal subunits, 50S, 30S. And in the ribosomal 30S subunit, we have the 16S ribosomal rRNA and the 21 proteins, which makes the 30S ribosomal subunit. And as you can see here, look into this picture. So this is the start codon and the stop codon. Let me show you a bigger picture. So this is the start codon and the stop codon. Where is that picture? So this is the Shine-Delgado sequence, and this is the initiation codon. And you can see the 30th ribosomal subunit first recognizes the Shine-Delgado sequence, base pairs with it, and then you can see, as you can see, it has also recognized the start codon, the AUG, and then after recognizing the AUG, it will start translating the polypeptide. So the AUG is for methionine, and once the start codon has been recognized, as you can see, the initiator tRNA, the tRNA containing methionine is brought into this portion and it recognizes the AUG. So this is from where we are starting our polypeptide synthesis, and this is the part upstream of the start codon, the Shine-Delgado sequence, which is necessary for the 30S subunit to bind with the mRNA and thus further recognize the start codon.